Thank you for joining my talk about targeting cognitive decline with repurposed drugs. That's dear to my heart because I'm a clinician. I want to prevent cognitive decline, and what we have at the moment are repurposed drugs. So that might be a solution to actually overcome dementia. But of course, I will start with a slide showing you that the population is aging. We are growing old. The number of 60 and 80-year-olds is increasing every year, and especially in Asia. And that's also the reason why I just started at NUS, the National University of Singapore. Of course, they see that the population is changing and we might want to prevent age-related diseases here. With the aging process, there is also a change in cognition. Namely, our cognition is declining during our life trajectory. Here you see a meta-regression analysis looking at global cognition measured by the MMSE, the Mini-Mental State Examination, over the life courses, and every dot are studies. And what you can see is that there's decline of MMSE around about the age of 40 onwards, a slight decline, and at the age of 70, there is a higher decline. And the green dotted line is the normal value, so 24 MMSE points and higher means normal, and lower it's abnormal. And what you can see is that there's a huge amount of populations at higher age who have abnormal global cognition. So knowing that we are aging, knowing that cognition is declining, we need to know what the risk factors are for example, incident dementia, and that's what we evaluated here. We used a huge data set of more than 400,000 individuals aged 65 years and older of a primary care data set. And what we wanted to know, what kind of risk factors can we identify already at age 65 and older, and are these risk factors being dependent on the age of the population? And what you can see in this slide is that hypertension, diabetes, hyperlipidemia, and stroke are highly associated with the incident dementia in individuals who are middle-aged, 65 to 70. However, this association is not present at oldest old populations, for example, at the age of 85 and onwards. So there is a different association of risk factors and dementia. And what you can also see, but that's quite logical, that heart failure is associated with incident dementia, and especially at younger age groups at the age of 65. So which means, of course, we don't know, is aging the driver, or is for dementia, or is aging driving the metabolic and cardiovascular diseases and then leading to dementia? So of course, we cannot disentangle that here. I would like to show you another studies looking at multimorbidity trajectories, also recently being published in Nature Communications by a colleague of mine from the Swedish National Study on Aging and Care cohort. And what they tried to do, they tried to predict over a 12-year period what the outcomes of individuals were at the age of 76. And they defined, of course, first the outcome, which is quite obvious, and that's death. But they looked at, okay, what kind of specific clusters can we find which are associated with certain outcomes at the age 6 years later or 12 years later. And what you can see, they define specific clusters as respiratory disease, cognition, of cognitive impairment, eye diseases, heart disease, psychiatric diseases. And what you can clearly see, the ones in the cognitive sensory impairment cluster, they're very likely to die. However, part of them are also going into the neuropsychiatric and respiratory disease cluster, which means these two disease clusters are linked to each other. If you look at heart disease, individuals with heart disease are very likely to die. However, they are also likely to have then cognitive impairment, which fits quite nicely with our study. And of course, if you then look 6 years later, so in total 12 years, these are associated then with cardiometabolic disease and of course a higher risk of death. So we are knowing that all the age-related diseases are associated with each other, but what we really need to know is why this happens. So, and then of course, Geroscience kicks in, or the biology of aging kicks in, because I'm a geriatrician and internal medicine specialist. We always look at higher functional levels. So we will measure cognition, we will measure functional status, we will look at comorbidities, we will write down if somebody has polypharmacy, and if they have a dog or a cat or whatever, what the lifestyle is. But we don't yet understand, I would say, or it's not implemented in clinical practice, that we use markers around there who could actually say how the biological aging process in these individuals are going. And we coined it a couple of years ago, biological geriatric assessment, which is an analogy of the comprehensive geriatric assessment we are using as geriatricians. And what you can see is that there are a couple of hallmarks of aging in that geriatric assessment, the biological one, and one is senescence. And I just want to show you in 2 more slides how these markers of aging are already associated. We have lots of knowledge with aging or age-related diseases. And I will focus on senescence because my group is focusing on that one. Here you see a systematic review where we looked at the association of senescence with chronological age in different organ systems. And what you can see on the horizontal axis are the different organ systems, and the vertical axis is the magnitude of the association of senescence with chronological age. And what you can clearly see is that, yes, most of the organs, we see that positive association with senescence. However, there are 2 organs really strikingly are more associated with some others, like the colon, and that's the pancreas and that's the brain. So we know that there is a high correlation of cellular senescence with chronological age in the brain and in the pancreas and all other organ systems. And we also know that senescence is associated with age-related diseases and also in the brain, for example, with dementia. This is another systematic review and meta-analysis we did, looking at all senescence markers and all age-related diseases in humans we were able to find. And one of them is the brain, that's depicted here. And all the blue dots are associations which are positive, so which means higher cellular senescence load with age-related diseases compared to the controls. And the red ones are not statistically significant. And what you can clearly see, so much blue, which means there is an established association of senescence and age-related diseases, including the brain, so including Alzheimer's disease, of other types of dementia. So knowing all this, of course, as clinicians, we want to act. So how do we make sure that somebody is aging gracefully, hopefully without age-related diseases, or can we actually slow down the aging trajectory? And this is a nice picture of the mother of Saudek, who made this picture really so gracefully, so strongly showing how somebody can age. And the aim of Geroscience is to expand health span and to decrease the period of within our lifetime with age-related diseases. So really expanding the health span, no age-related diseases, and then the steep decline towards death. If you think, oh, I've never heard about Geroscience, doesn't matter. Lots of clinicians do not know Geroscience yet because it's really an evolving speciality. And that's also being highlighted in this editorial, a quite nice one with the title Geroscience Coming of Age, and really highlighting there is so much lack of knowledge, but also so much opportunity. So if we think that aging is a driver for age-related diseases, could we then antagonize the aging process to prevent age-related diseases, to increase the health span, and to prevent that we have a long period of time with age-related diseases? And one of the factors which is necessary to do so, as clinicians, we need an ICD-10 code, an International Classifications of Diseases code, and we have one since 2019. And that's called the Aging-Related Disease Code, so XT9T. So we can already use it and has been approved already by the regulatory systems, which is very important. So then, of course, do we know why we age? And yes, we know it. And here you see the mechanisms of aging, the 9 hallmarks of aging. which are also influenced, and that's quite nicely with environmental factors. And I would like to highlight that we have these 9 hallmarks, for example, mitochondrial dysfunction or telomere attrition, stem cell exhaustion, epigenetic alteration, and then the nutrient sending dysregulation or cellular senescence. So many, and of course we are not there yet, so there might be much more around. But what we know is that with our lifestyle, we can already influence these 9 hallmarks of aging. So I will only show you one slide, but really highlighting how important physical activity and sedentary behavior is, and how important it is as clinicians to highlight that with our patients or with our clients. And I'm showing you here a couple of systematic reviews, a meta-analysis, and then an umbrella meta-analysis, which was recently accepted in the Lancet Healthy Longevity. But actually what we are showing here in thousands of individuals is that if somebody has a high physical activity, a low sedentary behavior, that is associated with positive outcome, which means survival, good activities of daily living, low frailty, no falls or low falls, good muscle strength, muscle power, and very importantly for this talk, good global cognition. So individuals with lots of physical activity, low sedentary behavior, they are more likely to have a good global cognition. So, and this is already something we can prescribe. Don't wait until we have the drugs, but really ask your individuals and do it yourself, be physically active. So read this systematic review. So what can we do with drugs, and why are repurposed drugs so important? And why might this be the way to go for maybe the next 5 years? Of course, we, and as, for example, internal medicine specialists, we know how drugs work. We have decades of experience, they're available, we know the mechanisms, and they are safe. And most of the times they are cheap or cheaper. And that's very important because everybody is aging at the moment, which means that we need a drug being affordable. Which, if it works, we should give it to lots of individuals or to everybody, or at least everybody should have the drug available if the drug is effective. So that's what I like for repurposed drugs, and we know the mechanism. So what we did in the past years, we looked at studies with a known mechanism, for example, how they influence proteostasis. And if these drugs were already tested to improve cognition, either in animals or in humans. Here you see the first systematic review showing the results of repurposing proteostasis-modifying drugs to improve cognition. On the left-hand side, you see all the drugs, and then in the middle, you see in which animal models it was done. And then on the right-hand side, you see the human trials. Everything green is a significant in the right direction, a positive effect, and the black ones are negative. And the X's indicated are trials ongoing. And what you can see, there are a couple of trials being tested already in animals, but also in humans, which is quite interesting. There's lots of green, which is also quite positive. For example, Rapalox, rapamycin, or lithium, or for example, doxycycline. So these are drugs with known mechanisms influencing proteostasis, which are now being tested or already have been tested with a positive results to modify cognition, especially to improve cognition. We then look at a huge number of drugs in the nutrient sending profile. What the results here are on the left-hand side, you see the drugs, then in the middle, you see the species, and then two different categories. That's studies who wanted to prevent cognitive decline or to cure cognitive impairment or dementia. So there's much more in terms of type of studies to cure than prevention. That's the first thing we see, and everything is red is negative, so no effect, and green is a positive effect, so improvement of cognition or curing of dementia. And what you can see is also that lots of studies are still in animals. So there are not many human trials yet. So which means that we should really, or not enough trials, we should gather together as clinicians and build these trial networks, working together to bring these trials on our plate and our daily plate, that as other specialties, for example, oncologists or cardiologists, they do trials always. Let's gather together and also do that within longevity medicine. That would be my goal for this meeting at least. But what you can see is that, yes, there's lots of negative, but I also indicated, I think, some very promising drugs in green, and you see insulin there, you see metformin, and you know that near Barzilai, Professor Barzilai is very busy with the TAME trial. So I think we already have evidence, yes, we can do trials, but what works, we don't know yet, and especially not to improve cognition. These are all repurposed drugs, but we could also, of course, look at novel compounds we find either often in nature, or we synthesize them. And one of the very interesting pathways is the GSK3 pathway, so the glycogen synthase kinase 3 pathway. And because we know that GSK3 activity, if that's high, it increases the A-beta production. And of course, there's lots of discussion about E-beta, but we know that E-beta is associated with cognitive decline and dementia. So the idea is, if we inhibit GSK3, the activity, that then we might could prevent cognitive decline or dementia. And we looked at compounds which could inhibit GSK3, and here you find some of the novel compounds, which are found in curcumin, for example, or they are found in sea cucumber, in lots of essential ingredients we already use in our food. So the question is, if you would give these either to humans or animals, would that improve cognition? And unluckily, there are no human studies yet. We just finished the search, but there is quite a lot of activity within the animal studies field. Here you see all the studies looking at GSK3 inhibitors to improve cognition, and everything circled with green is positive. So the majority of trials are positive. There are a couple which are unlikely to be positive, really neutral, and very neutral ones are the ones who tested GSK3 inhibitors in normal aging mice, which is of course then a pity, but you also see these were only 2 trials, whereas trying to cure cognitive impairment or dementia, there's much more around. So hopefully I convinced you today that yes, we understand, at least a little bit clinically, yes, we understand how cognitive decline looks like and how we can measure it, but that cognitive decline and changes in our brain are really associated with aging markers, and we can test like markers for cellular senescence. And we already have something very powerful in our hands, that physical activity, but we also should really move towards the field to repurpose drugs or to test new compounds. So if you would like to be engaged in such a network, please contact me. Or if you have any questions, please Twitter or contact me on LinkedIn or send me an email. I'm looking forward to it. Thank you very much.